This is Breakthroughs. I'm Erin Spain, Communications Director at Northwestern University Feinberg School of Medicine. A team of Northwestern scientists has come together from across disciplines to develop a new antibody test for COVID-19 that's designed for at-home use. This inexpensive, relatively painless test would allow people to collect their own blood with a finger prick and send samples to a lab through the mail to determine prior exposure to the virus. Dr. Elizabeth McNally is part of the team working on this test. She's a human geneticist, Northwestern medicine cardiologist, and the director for the Center for Genetic Medicine here at Feinberg. Thanks for joining me, Dr. McNally, and for being back on the show.
Oh, happy to be here and with what we hope is a really good test that will help us learn some good information about how this virus has spread.
I want to hear more about this test. The Northwestern team working on the antibody test includes biological anthropologists, pharmacists, scientists, physicians. Tell me first about this all-star team and how you all came together to create the tests.
Well, the process really started, I would tell you, March 14th, when most people know the story that we had hoped as a nation to have viral testing set up quickly. And it turned out we didn't. And so on February 29th was when the CDC and FDA told hospitals, well, you can try to develop your own test for the virus because essentially we don't have one for the country. So what happened was Northwestern Medicine quickly stood up its viral testing, and they had something up and going pretty quickly. But on March 14th, we got a call and said, we can do the test, but we don't have much capacity because there's lots of steps in this test that require hands-on skills. And so it just requires a lot. And a number of us in the research space recognize that, hey, we have those, not only we have the skills, we have the equipment, and we have the things needed to do it. So quite quickly, we extended the diagnostic test for the hospital where they normally do viral testing is under what's called CLIA certification. We got that extended to our research facility, specifically our NewSeq sequencing core, because they had all the capabilities and machinery to be able to do the viral PCR testing. That's for the acute virus. Took about a week to get that up and going and get all the certifications in place. And very quickly, even within that week, we realized, well, the next phase of testing, sure, we want to know where and when the virus is, but we also want to know where it's been. And the way you're going to do that is to test for antibodies. So antibodies develop in the bloodstream typically about 2 weeks after an infection, depends which type of antibody you're looking at. And so right away, we started to look into the process of what is it going to take to get antibody testing going more broadly, recognizing that because we couldn't test virus as rapidly as we wanted to, we would want this backward look to be able to know where the virus had been. And we discovered very quickly the same problems around viral testing, which was the materials you needed were in short supply. And so we put our heads together and we said, we contracted to get some of the viral protein made. We were able to rapidly move that in-house and get that set up and perform the ELISAs and be able to do it. But in the process of doing that, I had been working with Rich Tequila, who's in infectious diseases, and he said, you should talk to this guy, Tom McDade. He's done all the blood spot testing. So we reached out to Tom, and in like no time flat, we had groups working in parallel in Evanston, working in Chicago, making sure that not only that we could do the ELISAs, but that we could do them with high reproducibility. And it didn't matter whose hands were doing it, didn't matter whether it was in the Evanston lab or the Chicago lab, but that it had high reproducibility. So it really came together very quickly. And we were able to show that we could get the exact same results off serum testing that we could get off the dried blood spots. And so the real advantage of this is that serum, as you can gather, serum is just you get that off a blood draw, which requires having a person come to a place where there's a doctor or healthcare provider, nurse, someone who can draw that blood and collect it. The advantage of the dried blood spot is that it can be done in your own house. So you will receive a kit in the mail. It will have what's called a lancet, which is a little pointy object like a needle, and you poke that into your finger, you put a drop of blood on a card, and that can be safely mailed back to us, where we can then do the testing in the laboratory setting in a very highly quantitative and reproducible way to measure the amount of antibodies in the blood.
And this dried blood spot method, which was pioneered by who you mentioned, Northwestern anthropologist Thomas McDade, it's been used in the past. Tell me about that. This is something that he has used time and again.
Yeah, so dried blood spots are a very old technique that I was looking as Tom and I were working on this, he actually was citing references from 1963. So some of the earliest uses of blood spots were, I think people are familiar with, for example, when babies are born, they will take a spot of blood to do tests for different things, different genetic diseases that it's been used for, all sorts of things. Because you can test for proteins like antibodies, you can test for DNA in a blood spot, you can test for chemicals like different abnormal metabolites that might be in the bloodstream because of a genetic disease. So they've actually been used for a lot of things going back a long period of time because they're essentially a very simple system which can be widely applied. It just needs a drop of blood and a special card to collect the information on. And really importantly is they're safe to put in the mail. So that was the critical part of it, which is that you can put these in the mail very safely and send them without risk to the postal carrier or anybody else who might come in contact with it.
And this type of test, this more easy to use test, how could this promote more widespread testing?
Well, one of the things we've seen a couple of these early serology surveys come out, one from Los Angeles, New York City's had some, Santa Clara County in California. One of the things that we noticed in all of those is that it required people to come and be in contact with where the testing site was. So you have to leave your house during a pandemic and go to a place where there are a bunch of people wearing protective equipment, all gowned and masked, and you have to come in contact with them to give your blood sample in order to do that. So it became really clear that if you could have a test that you could, again, just send to the person in their house, it doesn't require them to leave their house to do the test, which we think is a huge advantage. Because if you're trying to do a survey of what's happening in the population, well, right now, only a certain few people are coming out of their house. So you really want to more broadly survey the population, including those people who don't want to leave their house right now.
Want this available, would it make sense for everybody to get this test, even people who've never had symptoms of COVID-19?
I think that's what we're learning from the process right now. We're just in the early phases of looking at smaller groups of people as we've been validating the test. We're now ready to do it more broadly. But in that validation, and based on what also has been seen in the New York City study, it's pretty clear there are people who develop antibodies to this that really had no symptoms at all. I think we all hope to be among those very lucky few that somehow you got exposed to it, you developed antibodies, and you might never have known you were sick. We've been hearing lots of stories about asymptomatic carriers, people who might even be shedding the virus and spreading it to other people unknowingly. And that's why we're all working hard to wear masks right now. We also know that a lot of people have been sick. They've gotten fever, some diarrhea, some cold symptoms, different things along those lines, but not sick enough to go to the hospital. And for some of those people, those symptoms have been, again, very, very mild. What we've learned most about and heard most about is the people who've become very sick from this virus. And we've all heard the devastating stories of how this has affected even families and different households in different ways. But that's the tip of the iceberg. We know that it's a small percent of people who become very, very sick like that. From a research perspective, we really want to start to understand the difference of who became really sick, why did they become really sick, and another way of looking at that same question is actually asking, why were some people protected? Why did some people have it so mildly? So being able to get a better cohort of people who developed antibodies but really weren't sick is going to teach us a lot of information about what it takes to be protected from this.
What comes next for this test? You mentioned it's still fairly early on. The first paper about the project was just released. Tell me about the next steps.
Yeah, so the next steps for us, we have two protocols that we hope to do. One will be to reach out to certain communities within the Chicagoland area, and we're trying to be selective about those communities, optimizing for getting a diverse sample, because we know there's been issues about how ancestry, for example, has played into who gets really sick and who may die from this. We've seen some disparities about that. We don't know whether that's driven by socioeconomic conditions, whether that's driven by genetic conditions. We don't know what's actually driving those disparities that we are seeing in who gets this and who gets very sick. So we're going to select some certain areas so that we can do some proper comparisons with a limited sample size to get started. The other piece we'd like to do is we have a large biobank here at Northwestern that's called NewGene, where we have DNA on about 15,000 people. And we have genetic information on about more than half of those individuals. So we're hoping to also reach out to those people who've previously participated in our biobank Because if we can get a sample from there and recognize some asymptomatic people as well as some symptomatic people, we already have the genetic information on those. So we can begin to ask that genetic question in a much quicker fashion to begin to say, well, these genes might be protective and these genes might be susceptible. And that'll help us guide better if you do get exposed, how careful you need to be going forward for your own health as well as the health of others.
And this is a solely Northwestern project. So many people from across the university tell me about that. How many different folks across Northwestern were involved?
Well, as I told you early on, a lot of different people from a lot of different departments. Our expertise is working with genetic studies, which is why you hear us talk about that and why we have an interest in that. But we also have a fantastic group here, Brian Mustansky's group and Rich DeQuilla, who have done a lot of work in virology, and Brian in particular working with reaching out to the community. And they've worked a lot with the HIV at-risk community, so they know a lot about reaching out to communities that way. And then, of course, Tom, who has all the dried blood spot expertise, and Alexis de Mondbrin, who's an assistant professor in pharmacology. She's been leading a lot of the hands-on work of really developing the assay and making sure it works. So it's a lot of different pieces and parts. We recognize we probably need even a bigger team, but we're working hard to get it done.
I want to go back to when you mentioned that NewSeq was part of this response to test those nasal swabs from symptomatic patients. What's happening right now with that project? Are we still assisting the healthcare system here at Feinberg?
Yes, we are. It's been ebbing and flowing as we've heard all the stories about viral testing. When viral testing first got set up, the major issue was that the nasal swabs themselves were not available. And I think many people have heard the story. That's because the major site where they're produced is actually Wuhan, China, and another facility in Italy, actually. So it was an unfortunate circumstance that nasal swabs were really quite limiting throughout the world. So that limited a lot of the testing that we could do early on. That's now been relieved, and we are able to get through that. And luckily, at the same time, the hospital does have some machines that actually can do the testing. So that's their first line approach right now, which is to use their standard clinical machines that are for this. And so the human parts of this are really serving as backup to when they don't have enough bandwidth on the machines, or sometimes the supplies for the machines have come in short availability at different points through this process. So right now they are able to do a lot of testing, and the capacity has gone considerably up. I think most of us in Illinois have recognized the governor has pushed really hard to increase our testing capacity. So that's gotten better, but the NewSeq facility is still helping out, serving as the backup to the hospital.
So when might people see these kits for the antibody tests in their own mailboxes? When do you think that could really happen?
Well, we're just waiting on our last approval, and then we will have a website set up. And right now, because we want to try to do good comparison between certain areas and neighborhoods, so we're going to have to limit it early on to what will satisfy the greatest use we can get out of the small number that we can do initially. And when I say small, it's still 5,000 that we'll do to start. So it's not that small. But on a population-based study, 5,000 is still considered small. So we're hoping to expand and to be able to do more, but that website will be a key place where people can enter in their information, and then we'll let people know whether they're accepted into the study.
Well, thank you so much for all this information. Is there anything else that you want to add about the project that you didn't get a chance to share?
Well, we do hope that it makes it more available to other people. Like I said, we're just getting started with this first study. The other piece of this is that look for it to change over time, because we're going to want to go back to even more people in the future. The more we can get, the better this study will actually be to be able to help answer these questions. We also expect it to change over time. The answer we get now as we survey through the month of May or June is going to look different than when we do this survey again in August and September. It's going to be very important to ask those different questions at different points in time, because we will get different answers. The assay that we're using is one that was set up by a group at Mount Sinai Hospital in New York. Dr. Florian Kramer's group is the one who set it up. And I have to say, they did a terrific job establishing an assay and then working really quickly to make the information available to other people so that it could be replicated. And without that, we really couldn't have gotten it off the ground. So I hope people recognize that a lot of that was actually done in academic research labs, and we were able to move it out very quickly and be able to do that. So this is really a big effort going on that's involving many different pieces and parts, and a lot of really good information sharing to make it happen quickly.
Well, thank you for your work with this project, and we can't wait to talk to you in the months to come to understand what you found out.
Yes, we're looking forward to seeing what those results look like and how they change over time.
Thanks, Dr. McNally, for joining me today.
Great. Thank you. Be well.
Be sure to subscribe to the Breakthroughs podcast so you can stay up to date on the latest COVID-19 research from Feinberg faculty.